Morning, everyone. In 2013, I completed a postdoctoral fellowship with Dr. Mary Rice, former curator of worms here at the museum and the founder of the Smithsonian Marine Station in Fort Pierce, Florida. Early this year, I completed a second postdoctoral fellowship at the Smithsonian Tropical Research Institute in Panama. And now I'm back at the Marine Station to manage the future course of research in the Life Histories program and carry on the great legacy of Mary Rice. The tools I use for my research include developmental biology, different types of microscopes, and the expression of genes during early animal development. These tools allow us to not only gain insights on the origin of species, but also the movement of species around the planet, especially in the oceans. In the Life Histories program, I use them to study a group of sipunculin marine worms, like the magnificent specimen on the screen in front of you. This is the adult reproductive stage of Fasciculosoma perluscens. Like many invertebrates, it has a very complex life history that includes different larval stages. The first larva is called a trochophore larva, and the second larva is called a pelagosphaera, which is unique among all animals. The life history of Fasciculosoma begins here with the very first cell division of a fertilized egg, dividing a single cell into two cells, eventually the embryo, and then a series of larval stages. Here. On the far left is the 2-day-old trochophore larva. On the far right is the 4-day-old pelagosphaera larva. In the middle is one of several different transitional species, or larval forms, excuse me. These images were produced by a confocal laser scanning microscope. Each of the blue dots you see is DNA in the nucleus of a single cell, and the yellow color is filamentous proteins that are used for swimming, feeding, and conduction of the nervous system. The confocal microscope enables me to look into and see the internal structure of microscopic organisms. For perspective, you could stack about 5 of these larvae along the thickness of your fingernail. I'm also using the confocal to study the evolution and development of the nervous system. On the left is one of these transitional larval forms, but now with a third color in red, which I pulled out to the right. This is a component of the central nervous system of this particular species. From the top down, it shows a group of cells that represent the brain and the neuropil, 2 lateral nerves running along each side of the mouth, and a group of cells branching along the ventral nerve cord. I'm also using it to look at some of the large pelagosphaera larvae from the open ocean, in particular the Gulf Stream that runs along the southeast coast of Florida and up around the North Atlantic. By looking at different organ systems and labeling them, such as the position of DNA showing cells, the nerves, and the musculature, we're now getting information on morphology, development, function, and ultimately the behavior of these larval forms. Each one of these images represents one anterior or head region of a single larva. This particular beautiful one right here, the yellow papillated pelagosphaera, shown in lateral view on the left and ventral view on the right. From the top of the screen, we see the head region, followed by a bright white circlet of cilia that represents the metatroch, which is kind of like an airplane propeller that allows the larva to swim through the water. And the large yellow-orange trunk in the bottom region houses a number of different organ systems, including the reproductive system, digestive system, excretory system, blood cells, and more. Now, for more than 40 years, we've not been able to identify which species this belongs to. But now with the advent of molecular barcoding, we're gaining molecular fingerprints that allow us to match larval forms with adult forms, like this, Spitosiphon laevis, one of my favorite sipunculins. It's found from Florida through South America in coral reef structure. For the last 3 years, I've been down in Panama, and I've been collecting, barcoding, and imaging hundreds of different larval forms to try to finally get the identity of these animals. And these include very special species, including animals like this. This is the bipinnaria of a sea star or starfish, shown in bright field microscopy on the left and dark field on the right. Mueller's larva of a platyhelminth flatworm with ciliated lobes for arms and the classic black pigment spot showing the position of photoreceptor cells or eyes, and the tornaria larva of an acorn worm that some say looked like the helmet of Luke Skywalker's archenemy, Darth Vader. More recently, I've been using genomics to build large inventories of genes that are known to have major roles in building the body plans of animals across the tree of life. And I'm doing this for sipunculin worms. From these, I have select groups of special interest, including genes that pattern the digestive system, genes that build the segments of insects, polychaetes, and vertebrates, and genes that are important in the development of the nervous system that likely play a major role in all animals, including jellyfish and you and I. From this list, I can take a single gene of DNA, transcribe a piece of DNA in the form of RNA, introduce it into the cells and tissues of the organisms during development, allow it to bind with the exact copy of the gene that the animal is producing during development, and then add some chemistry which causes a blue color showing you exactly when and where this gene is being expressed during development. In this case, FOXA is expressed in the pharynx or the foregut and the intestine during development of Fasciculosoma cryptum. And we're now able to start to use gene expression in cypunculins to compare that with all of the model systems that have been discovered or have been studied for decades. In the future, I'm hoping to collect the cell types and genes that are used to construct one of the most fascinating larval types on the planet, the large cypunculin pelagosphere of the open ocean. To my mentors and advisors, to the very efficient staff at the Marine Station, and to each of you here this morning, thank you very much for your time.
Are you working mainly on in Florida or worldwide, or where's your study area?
I've been working both in Panama and Florida, and that's right now that is my study area, but I hope to expand that. These worms are found worldwide, from the poles to the equator, from the near shore all the way to the abyssal plains. Some of them even down below 6,000 meters, so it's wide open. So my primary interest is in the evolution of animal form, especially body plans and the radiation of animals throughout the history of life. For more than a century, most of the attention has been given to 5 or 6 main model organisms: C. elegans, the nematode, the fruit fly, the sea urchin, and so on. In my field of evolutionary developmental biology, or EVO-DEVO, we're starting a trend to fill in the gaps in getting a better representation of all the different animal forms, especially the obscure animals that nobody has really studied and are difficult to study. So the fruit fly and the sea urchin, although there's been great progress and they've contributed a lot to biomedical research, they don't really represent the diversity of animals on Earth. So these new understudied and new non-model organisms are starting to become very important, and I'm making progress on one particular group of worms where nobody has done that, and building upon a legacy that has a lot of information on reproduction and development. So my goal is not really to promote cypunculins, but to use cypunculins as a model to understand much more diversity.
Could you please repeat the question?
Michael, have you submitted some of your images to photo contests? There's one image that's in a gallery somewhere in the museum for the staff photo contest. 2 of the images have found honorable mentions in the Nikon Small World contest, and one in the Olympus contest, and more are on the way. Thank you.